Okay, so welcome to this next video in which we are discussing transcription factors. Okay, so we're currently in the process of discussing the different motifs which you can find within transcription factors. So we've discussed the helix-turn-helix motif, we've discussed the homeodomain, which is a specific motif that comes under the class of helix-turn-helix motifs. And now what we're going to move on to is the zinc finger motifs. Okay, right. So zinc finger motifs, everyone hopes that it's going to look like a finger in some way, but the name zinc finger motif was given a long time ago, when the only ones which were known actually did look like fingers, at least when people drew diagrams of them. Okay, the zinc finger motifs is now an absolutely massive great family of motifs that you can find within transcription factors, and really the only characterizing feature that they all have is that they have a zinc cation within them. Okay, so do not expect these to look like fingers in any way. The characteristic feature that they have is that they have a zinc cation which is involved in holding the full thing together. Okay, so I am going to give you a specific example of a zinc finger motif, because there are loads of different zinc finger motifs. But I'm going to give you a specific example, and the one that I'm going to talk about is the Cis2 His2 motif. Okay, and this is because the amino acids which are going to bind to the zinc cation are going to be 2 cysteine residues and 2 histidine residues, and we'll talk about what histidine and cysteine residues look like in a moment. Okay, so I will firstly draw you a small picture of this, and then I think we'll try and draw a bigger picture of this where we actually show the interactions between the histidine residues, the cysteine residues, and the zinc cation. Okay, right. So, firstly, let's begin with the amino terminus. Okay, so here is the amino terminus. Okay, then there could be an additional domain here. Then we're going to go into what's known as a beta strand. Now, what is a beta strand? Well, a beta strand is a beautifully straight portion of the polypeptide that is capable of interacting with other beautifully straight portions of the polypeptide. And the way that they will interact is by forming hydrogen bonds between the groups within the peptide links of parallel strands, basically. Okay, so here is one beta strand here, and it's oriented in this direction, i.e. the polypeptide progresses in this direction. Then you'll have a loop taking you into the next beta strand, which is anti-parallel to this first one, and there will be nice hydrogen bonds forming between these two beautifully parallel strands. Okay, then that's the end of the beta strands. Then you have an alpha helix over here. Okay, so up we go with the alpha helix, like so. And then you'll finish there, and you'll go into the carboxylic acid terminus over here, after potentially a few more interesting domains. Now, that's all well and good, but where is the zinc atom? Well, I haven't shown it yet. Okay, basically, we have two separate structures here at the moment. We have this small little beta pleated sheet here containing just 2 beta strands, and then we have the alpha helix. The zinc cation is basically going to sit in the middle of these two, and it's going to hold the two together, basically. Okay, now there are 4 amino acids that are going to interact with this. 2 of them are going to be in the alpha helix, and these are both going to be histidine residues, which for now I'll just abbreviate as H. Okay, and these are going to form little bonds, little electrostatic interactions. Okay, they're not covalent bonds as such. Okay, but they're coordinate bonds. Okay, they involve a nitrogen atom giving its electrons towards the zinc cation. Okay, then we've got 2 cysteine residues, one which is in the loop between the 2 beta strands, and the other which is in this first beta strand, and these are going to form covalent bonds with the zinc cation there. Okay, right, so that's how the zinc cation is involved in the structure. It's holding these two beta strands together with the alpha helix here. So these are alpha helices. Well, this is our alpha helix, and these are beta strands. Okay, right. So now let's discuss the structure of cysteine residues and the structure of histidine residues in a bit more detail, and then we'll try and draw a bigger picture where we have the cysteine residues and the histidine residues drawn out in full and interacting with the zinc cation. So let's start with the structure of a cysteine residue then. Okay, so we'll start with the core amino acid structure. So here is the amino group. Then we've got the alpha carbon with a hydrogen coming off it, and here is the carboxylic acid group. And we are drawing an amino acid residue rather than the free amino acid. So we're drawing this amino acid as though it's bound to an amino acid prior to it and also an amino acid after it. Okay, and then the R group of a cysteine residue consists of a methylene group, like so, with a phyl group coming off it. Okay, so this is a cysteine residue. Okay, then we also want to show a histidine residue. But before we go on to histidine residues, let's just finish cysteine residues. This group that we've got here, which is a sulfur atom bound to a hydrogen atom, this is called a phyl group. Now, phyl groups have very similar chemical properties to alcohol groups, okay? And that's because sulfur is in the same group at the periodic table as oxygen, and therefore has similar chemical properties to oxygen. Now, if that was an oxygen, we'd call that an alcohol group. Okay, we've replaced it with a similar atom to oxygen, so we'd expect this phyl group then to have similar chemical properties to an alcohol group. Okay, right. So now let's draw the structure of a histidine residue. So once again, we'll start by drawing the core amino acid structure. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, and here's the carboxylic acid group. And then the R group of a histidine residue consists of a methylene group, like so, with something that we've seen already, with an imidazole ring coming off it. Okay, now we're going to draw the imidazole ring in full, basically. Okay, so here are those two carbons that are at the base with a double bond between them. Then we've got two nitrogen atoms, like so, and then we've got a double bond between one of these nitrogen atoms and the carbon, and a single bond between the other nitrogen atom and the carbon. Okay, like so. And then this nitrogen down here will have a hydrogen coming off it. This nitrogen here, this is going to have a lone pair of electrons. Well, in fact, both of them are going to have lone pairs of electrons. But it's the lone pair of electrons coming off this nitrogen which are going to interact with the zinc atom. Okay, right. So this is the structure then of a histidine residue. Okay, so let's now draw the structure of this cis2 his2 motif, which is a zinc finger motif, in more detail. Okay, so we'll draw this picture bigger now. So, we'll just show the R groups, we won't show the full amino acid structure. Okay, so here is the alpha helix, and then coming off it is going to be one of these histidine residues. Okay, I'm going to rotate the imidazole ring round so that the nitrogen is facing the base. Okay, so we'll have it like this instead now. So here is a nitrogen coming off here with a hydrogen. Here's a carbon, then we've got a nitrogen down here, double bound to that. Oh, and we should have had a hydrogen coming off there, I do apologise for that, and a hydrogen coming off there, in fact, since I'm drawing a molecular formula. I've got too used to drawing skeletal formulae. Okay, so there's our histidine residue, and this nitrogen now is going to form a nice interaction with our zinc cation here. Okay, so that's going to form a little bond there. So we'll colour that in green here. Okay, right. I'll also colour in the zinc cation in red here. Okay, then the alpha helix will continue on. Okay, so we'll go down a little bit more, and then we'll have another histidine residue coming off here. Okay, so here's its methylene group, and now I'll draw it this way up this time, so that the nitrogen is facing the zinc cation up there. Okay, here is the other nitrogen, Here's a carbon, here's a nitrogen. There's a double bond, another double bond. Okay, so I'm just copying this structure out here. The hydrogen that comes off that carbon, the hydrogen that comes off that nitrogen. Then the lone pair of electrons from this nitrogen is now going to form a nice interaction with the positively charged zinc cation up there. Okay, so this is going to form a coordinate bond there. Okay, then the alpha helix will continue, so I'll colour this alpha helix in blue here. Okay, so they'll continue on to loop round into these beta strands in a moment. Okay, so we'll continue it on a little bit further, and then we'll bring it round to form these beta strands. Okay, and I'm going to have to make sure that this is going to fit in. Okay, so let's finish the alpha helix being coloured in blue here. Okay, right, so firstly I think we probably should begin by showing the cysteine residues to make sure that we actually get them to fit in. So here is the R group of a cysteine residue, here's the methylene group, and then the sulfur atom is going to be bound by covalent bond to the zinc cation. So it's going to have given up its hydrogen atom and it's now going to be bound to the zinc cation instead. Similarly, for this second one, the sulfur atom will once again be covalently bound to the zinc cation. Okay, so two more interactions there. And then, here is our beta strand here. Here's the amino terminus here, with potentially more domains prior to it. And then we have our other beta strand here. Okay, so these two strands in red, these are beta strands. One going up like that, and the other coming down like that. Okay, and then, prior to the start of this alpha helix, we then have the amino group with potentially a few domains in between the alpha helix and the amino group. Okay, right, so this is our cis2 his2 motif. Okay, right, so that's it in more detail. So now we've finished zinc finger motifs, well and truly. We're now going to look at two final motifs that I'm going to talk about, which are leucine zipper motifs and Helix-Loop-Helix motifs. Now both of these are different from the other ones that we've talked about so far. In fact, I could probably fit them in down here. Okay, and the main difference that they have is we've talked about how transcription factors often dimerize. Okay, but the ones that we've seen so far, the helix-turn-helix motifs, the homeodomains, and the zinc finger motifs, they have other domains that are involved in, or rather the transcription factors which contain these motifs have other domains which are involved in the dimerization of the transcription factor, whereas the leucine zipper domains and the helix-loop-helix domains, those are actually going to be involved in the dimerization of the transcription factors which contain them themselves. And in fact, I think I will get another piece of paper. Okay, so let's now talk firstly about leucine zipper motifs. Okay, so this is a large category of transcription factors which have leucine zipper motifs within them. Okay, and a leucine zipper is basically an alpha helix which is going to dimerize with another transcription factor which contains one of these alpha helices, and the alpha helices will coil around one another, okay, in a distinctive manner. So, basically, a leucine zipper transcription factor will have this sort of a domain within it. It's going to have an amino group, then potentially with some other interesting domains prior to this alpha helix here. Okay, so here is this alpha helix. Then the alpha helix will finish. You'll then have other interesting domains potentially, and then the carboxylic acid group here. Okay, so this alpha helix is the key thing. Now, the reason it's called a leucine zipper is because these alpha helices are generally very rich in the amino acid leucine. So let me show you the structure of the amino acid residue for leucine. So here's the amino group, here's the alpha carbon with a hydrogen coming off it, here's the carboxylic acid group as though it's bound to the amino group of the next amino acid along, and then the R group of leucine consists of a methylene group like so, with a carbon coming off it which then has two methyl groups coming off it, like so, and then a hydrogen coming off it. So this R group of the leucine here is extremely hydrophobic, because if we look at this structure, we've got carbon atoms bound to carbon atoms bound to hydrogen atoms. None of those bonds are polar. Everything's got the same electronegativity roughly, so it's all extremely neutral. So you've got loads of these neutral structures sticking off the side. This is why leucine zipper domains tend to aggregate together, because if we bring another one of these here, which has also got lots of these leucine residues sticking off the side, they're going to be forced to aggregate together so that water doesn't have to interact with these hydrophobic leucine residues, basically. Okay, so hydrophobic interactions are going to hold these leucine zipper transcription factors together. So, that's why the leucine zipper domain is involved in dimerizing these together. They also have a remarkable way of binding to DNA, so I want to show this. Okay, so once again we'll show the DNA, and this time it's best to show it on the side, like so. Okay, so here is the second strand splitting us into the minor and major grooves, like so. Okay, and basically these alpha helices, which are the leucine zipper motifs of these transcription factors, are going to sit in between the, well, they're going to sit within the major grooves. So we've got one major groove here, okay, and this is the way they're going to dimerize. So here is one of these leucine zipper motifs of a transcription factor. Okay, and it's going, as you can see, within this major groove. Now, the other is going to go in this minor groove, because even though we've got a, sorry, it's going to go in this major groove here. Okay, this is a minor groove here, facing us, but if we were on the other strand side of the DNA, there would be a major groove going down like this, and that transcription factor leucine zipper domain is going to go down in that, so it's going to go behind the DNA here, so we're going to temporarily lose it, and it's going to go in this major groove here, and the two alpha helices here are coiled together, okay, interacting because they have these two leucine, well, they have lots of leucines within them. So, this is how these two alpha helices of the two leucine zipper motif containing transcription factors, often just called leucine zipper transcription factors, are going to bind to the DNA. Okay, and they're going to bind necessarily as a dimer, basically, because the leucine zipper motif itself dimerizes. Okay, right, so that's how leucine zippers work. Finally, the final motif we want to talk about is what's known as a helix-loop-helix motif. Okay, and this is similar to a helix-turn-helix motif, but the difference between helix-loop-helix motifs and helix-turn-helix motifs is that in helix-turn-helix motifs you had a helix followed by a turn followed by a helix. Now that loop between the two helices was tiny, and therefore it's called a turn rather than a loop. Okay, whereas in the case of helix-loop-helix motifs you have a much bigger loop between the two alpha helices. Okay, so basically these transcription factors look like so. You have the amino terminus here, okay, then you might have more domains in between the amino terminus and then the start of the helix-loop-helix motif. So here is the start of the helix-loop-helix motif. You start off with what's known as the long alpha helix. Okay, so here we have our long alpha helix. Okay, and I will colour in the long alpha helix in blue here. Okay, then you're going to have a large loop between the long alpha helix and then the next alpha helix, which is the short alpha helix. So we have now a nice long linker separating the long alpha helix and then the short alpha helix here. And then of course we can have more domains, and then we'll finish off with the carboxylic acid terminus here. So this now is the helix-loop-helix motif that is present within many different transcription factors. Okay, so we have our large looper or loop here in green. So this is our loop, and in vivid purple here, this is the short alpha helix. Okay, so that's the structure of a helix-loop-helix motif. Now these also dimerize, okay, and they dimerize in a very similar way to the leucine zipper transcription factors. Okay, so once again we'll draw a DNA strand because I'll show the two helix-loop-helix motifs dimerized as they are bound to the DNA. Okay, so here is our DNA, like so. Right, so you've got a major groove here, a major groove here. Okay, in fact, actually, no, that's a different way of looking at it. We'll have the major groove here, so we'll say that this is looping forward because you can look at this in two ways. Okay, so this is a minor groove facing us, this is a major groove here facing us. I think that's the best way of doing this. That's the way we did it on the picture above. Okay, so what we're then going to have is we're going to have the C-terminus of our helix-loop-helix motif here. We'll have the short alpha helix here. Okay, we then have the looper here, or the loop rather. I don't know why I'm calling it a looper. I'm mixing loop and linker together. Okay, and this is then going to come in front of that strand, which will hopefully give you a three-dimensional understanding of how this picture is working. And then here is the N-terminus. Okay, so let's colour that in blue here. So that's the alpha helix, the long alpha helix. Then we have the loop in green here. And then we have our short alpha helix in purple there. And it's going to dimerize with another one of these. So here's the C-terminus. Then I'll just do it in colours this time. So then we've got the short alpha helix. Then we've got the linker here, or the loop, like so. And then the other long alpha helix will interact and coil with this alpha helix here, and that's why they dimerize, and then it will go behind this strand here in the major groove on the other side of the DNA. So if we were looking from within the piece of paper up out at this piece of DNA, this would be a major groove. It's a minor groove on our side, but on the other side it's a major groove, and it's going down in that major groove. Okay, right, so that then is the structure of a helix-loop-helix motif and how they dimerize together to dimerize the two transcription factors which contain these helix-loop-helix motifs together. Okay, so that now concludes our discussion of transcription factors.